Good morning to all of you. I just continue with the topic on carcinogenesis, which I started last week. Hope you're all safe. Spend your time usefully. Don't waste it. Please follow the instructions that would be posted to you on WhatsApp. So continuing with the topic which I had taken last week, all of you know that this is a repetition. You've already learnt it from Dr. Rajeshri, but I'm just repeating, stressing on certain important aspects where probably if all of us know, we can prevent these carcinogens acting on us. Okay? Let me begin with, starting with carcinogenesis part 2. Objectives of this session, it's a repetition class, right? So I'm dealing with naturally occurring carcinogens and miscellaneous carcinogens. What aspects about the source and mechanism of their action with a review of the previous class. You all remember this classification? Here it may not be visible properly, but your textbooks have this classification. It's an important main question for you in the examination. So you should know to classify chemical carcinogens. So two broad categories: direct acting carcinogens and indirect acting carcinogens. I just repeat some of the important aspects here. Direct acting carcinogens are chemicals. They do not require a metabolic conversion to become carcinogenic, but they are weak carcinogens. Okay, drugs, example. So which are the carcinogens in this group? See, the list is given here: cyclophosphamide, chlorambucil, busulfone. You know they're all anti-cancer drugs. So beta, propiolactone and epoxides. So second group we have acylating agents, acetyl imidazole and dimethyl carbamyl chloride. Okay, so direct acting carcinogens. Then alkylating agents are mechanism of action. If you want to see here, they contain electron deficient atoms. So they react with electron rich atoms in DNA. Thereby they injure the normal cells and not just destroy the cancer cells. So destroying the cancer cells, they also damage the normal cells. That's why the secondary tumors like development of solid and hematological malignancies, secondary to these drugs which are used. Now the indirect acting agents, indirect acting carcinogens. Why are they called as indirect acting? Because they require metabolic activation. They need to be converted into an ultimate carcinogen. Example is polycyclic aromatic hydrocarbons. So when we say polycyclic aromatic hydrocarbons, I'm sure all of you know the main sources of polycyclic aromatic hydrocarbon. So the important carcinogens there are benzopyrene. So we say 3,4-benzopyrene, 3-methylcholantharine, dibenzanthracene. Source, remember the previous class? We were talking about chimney sweeps. There was a cute boy who was carrying a brush with him and who was totally discolored. Then cigarette smoke, because it's a high temperature combustion of tobacco that produces this. Do you remember the smoked foods? Meat, fish and all that. So all these are the sources of, or the main carcinogen is polycyclic aromatic hydrocarbon in them. So they come under indirectly acting agents. Now, so let's move on to the next one. What can you see here? The pictures are so clear. Groundnut. Yes, again, whole groundnut with the shell. Then maize. Do they look healthy? They look diseased. That means there's fungus that has grown on them. So molds that grow on cereals, nuts and all that. So this all depends on the way they are stored. Okay. So these are the molds which you commonly encounter. I'm sure all of you have seen. You just open a few slices of bread and then keep it open in the room temperature or keep it in a box closed. Have you observed? Fungus growing on it. It looks black also, probably that is Aspergillus niger. So millions of people are exposed to. So these are aflatoxins. They're produced by Aspergillus. Aspergillus flavors, they say, molds. Where are these present? They're present in the air. So Aspergillus, as you know, in immunocompromised individuals, so it enters the lungs by inhalation and it stays in the tubercular cavity, forms a ball of fungus, aspergilloma. So this is an opportunistic infection, of course, in humans, but see how it is contaminated. All these are contaminated by aflatoxins. So it is the aflatoxin B1, a derivative which is genotoxic hepatocarcinogen. It produces hepatocellular carcinoma. So one of the etiological factors or risk factors for hepatocellular carcinoma, we call it as hepatoma, or a common man calls it as liver cancer. In addition, it also causes impaired immune function and also malnutrition, or people with immune compromised people, they're prone for this. So this is how the fungus appears microscopically. Can you make out this hyphae? They septate the branching. There's a small branch you can make out here. The branch at acute angles. They septate. And the fruiting body, which is very well shown here. So this is seen in culture smears. And sometimes even in histopathology we do see this. This is likened to or compared to the appearance of this. What is this? A holy sprinkler. Okay. So this is Aspergillus, the microscopic appearance. Okay, aflatoxins. What happens to these aflatoxins? B1, we said aflatoxin B1. So that is metabolized by cytochrome P450, exactly like how the polycyclic aromatic hydrocarbons get converted to the ultimate carcinogen, how epoxides are produced. So aflatoxin B1 is metabolized by cytochrome P50 to a reactive intermediate B1 to 8 and 9. That is, epoxides are formed. These epoxides we know are electrophilic and have electron deficient atoms. So they react with the proteins of the cell and the nucleic acids, DNA and RNA. So when they bind to the liver cell DNA, results in adducts. And in addition, you should remember aflatoxins are known to produce mutation of P53 gene also. So when we talk about hepatocellular carcinoma, aflatoxins producing hepatocellular carcinoma, just remember polyvinyl chloride also, which is also one of the etiological factors for not hepatocellular carcinoma, for angiosarcoma that occurs in the liver. So this is another malignant sarcoma that occurs in the liver. So it is the polyvinyl chloride, a monomeric form, which induces that, which is a risk factor. So who are the people who are exposed to this or who are at risk? People who work in plastic industries. See, all these are PVC pipes. We call them as PVC pipes. So plastic, angiosarcoma, malignant sarcoma of the blood vessels, arising from blood vessels. Liver is one of the common sites. Soft tissues and breast. It can occur at 3 places. See, we are moving on to aromatic amines. We are moving on to aromatic amines and azodyes. So aromatic amines are beta-naphthylamine. See, aniline dye and rubber industry. What are these dyes which are used? Food may look very attractive and colorful with these artificial colors or dyes which are added. Please avoid eating such foodstuffs. So butter yellow is one, which is added to butter and margarine to give an yellow color to it. Butter looks very attractive, so we tend to buy it. Then scarlet red. Foodstuffs with red color, especially the cherries. Tutti frutti, how much of color is added to it. Better avoid all these foods which have lot of colors, artificial colors which are added to it. So what is the mechanism of action then? All these dyes, especially the naphthylamine and all which we discussed, so they are mainly metabolized in the liver. So what happens in the liver with aniline dyes and all that, naphthylamine? So they are converted into active carcinogens there. But important factors, they detoxified immediately by conjugation with glucuronic acid which is there in the liver. See, a carcinogen is metabolized, converted into an active form, but immediately it is detoxified because of the glucuronic acid. Then what happens? It has to be excreted. So the paradox here is, when it is being excreted, it is deconjugated in the urinary tract by glucuronidase, an enzyme which is there. Very bad, sad. So now the urothelium is exposed to the reactive hydroxylamines. So where does the malignancy occur now? So it occurs in the bladder. So we say bladder malignancy, bladder carcinoma. What type of carcinoma occurs in the bladder? Just give a minute thought for that, seconds. What is the lining epithelium of the bladder? All of you know it is transitional epithelium. Okay, what is a benign tumor that occurs from this? A transitional cell papilloma. So what is the malignant counterpart? Transitional cell carcinoma. We call it as urothelial carcinoma now. And transitional papilloma. We don't call anything as papilloma now. Even that is considered as grade 0 malignant. So that also comes under urothelial malignancy. Look at this bottle. You're all familiar. You've done this test using this chemical to detect blood and urine. First year biochemistry you have done. Now second year also you're doing it. This is a test that is banned because benzidine we know is carcinogenic. Benzidine glacial acetic acid test. So you should never handle it. Okay, that also is one of the etiological factors for urinary bladder cancer. So nowadays we use urinary reagent strips to test all this. Or there is orthotolidine test or gum-goac test that could be used to detect blood in or presence of RBCs and urine or even stool sample. So better to avoid this test, benzidine test. So this slide again tells us that aromatic amines and azotides, the beta-naphthylamine which we were talking about, which is used in aniline dye and rubber industry. So workers in these industries have a high risk of developing bladder malignancy. This is a recap. Okay. Tobacco, betel nut, chewing. Okay. So tobacco, when we say tobacco, we know that smokeless tobacco also. Smoking is through BD, cigarettes, and what is this here? See, reverse chutta smoking. So burning end is kept inside. These are etiological agents for carcinoma of the oral cavity. BD, cigarettes. You can see all this, all pan parag and all that. Nice, shining strips which probably decorate all the shops on the roadside when you see. So kidney cancer. Just have a look at all this. So pan is also one of the etiological factors, which is, so when pan is kept as a quid in the oral cavity for a very long time, so it causes chronic irritation there. And that too pan with tobacco, that is more dangerous. All this, the pan, gutka and all that. So initially they cause submucous fibrosis. You have seen the oral cavity with a nice smooth lining of the mucous membrane. Initially it causes puckering, fibrosis of the mucosa. So later it is dysplasia and development of malignancy. So what malignancy develops? What's the most common? Very simple to answer if you know histology. What is the lining epithelium there? It is stratified squamous epithelium, non-keratinizing. All mucosas are lined by stratified squamous non-keratinizing epithelium. So it is squamous cell carcinoma. So this is another, probably we can say a physical agent causing carcinoma. Anybody has seen this? We have one kangri, this is called as kangri, kept in our museum. So if you have visited Kashmir or if you're a Kashmiri, so you'll be knowing about the usage of this. So there is an earthen pot here inside which contains burning charcoal, and this is kept in a basket made of cane or bamboo, whatever, cane, yeah, very nice. So very cold weather, so to keep themselves warm, Kashmiris use this. So they keep it either on the abdomen, cover it, or keep it between the thighs or between the legs, wherever it's they find it comfortable. So what happens? It's the heat, you know, that constantly irritates the skin. What has happened here? This is on the thigh, you can see a lesion, so which is a fungating, ulceroproliferative lesion here. So what malignancy is this? This is not a simple ulcer, because you know to differentiate a benign ulcer from a malignant ulcer. It's got a rolled, everted edges also. Not typically a cauliflower. Okay. This is squamous cell carcinoma of the skin of the thigh. Hope you people are not sleeping. I'm not able to see you. Yes. So naturally occurring products, we discussed some of them. Aflatoxin. So please avoid eating such stuff. You may not eat, but if you see people eating such grains and all that with molds, please tell them not to eat and then replace that with a better one for them. Because this Aspergillus flavus grows in stored grains and plants, especially rainy season and all, if it becomes wet. So other naturally occurring products are actinomycin D, mitomycin C, saffrole, betel nuts. We dealt with betel nuts. So miscellaneous agents, vinyl chloride. Remember that picture, the PVC pipe, then angiosarcoma of the liver. Then asbestos. People who work in asbestos industries, where they make sheets and all other products of asbestos. So they're prone to develop first a pneumoconiosis, occupational disorder, that lung disease, that is asbestosis. So after asbestosis, they're prone for bronchogenic carcinoma and mesothelioma. Another slide, I'll just tell you that about it in detail. Then arsenical compounds. So they cause epidermal hyperplasia and basal cell carcinoma. Metals, just written few metals here, nickel, lead, cobalt, cadmium, chromium, everything. They are the causative factors or implicated in the genesis of lung carcinoma. See, something interesting here, I've just put it in red. Saccharine and cyclamates. Do you know about them? They're the artificial sweeteners. No, diabetics, instead of using sugar, they use it. It's not advocated because studies have shown that they induce carcinoma in experimental animals. So best artificial, I should say natural sweeteners to be used by diabetics, I tell you, are stevia. It's a plant. So stevia powder is available. Then monk sugar. Two things, they're supposed to be very good, naturally occurring, not the synthetic ones. All the other sucralose, even what comes as brands like Splenda and all that, they're also supposed to be hazardous. So they have an action on the gut microorganisms, lactobacilli. So they totally reduce the number of this bacteria. So they're all helpful bacteria which need to be there in the gut. So better to avoid artificial sweeteners. See, there's a nice picture here, please have a look at it. This is a section from the lung microscopy. stained with hematoxylin and eosin. But nowhere you can say it is lung because you're not seeing alveoli. So the lung has undergone fibrosis. There's a disease process. There are probably some granulomas. I can make out. See the epithelioid cells, footprint-like nucleus. So here it is very well seen, even here. Okay, whatever it is. We are able to see certain structures which are brownish, yellowish brown in color. And look at the shape. It's got an elongated central part with two bulbous ends. So what does it resemble? You may call it a bone or whatever, but this is resembling a dumbbell. Okay, so there are structures which resemble dumbbell. Simile. One simile today we used previously was a holy water sprinkler. Now we are using a second simile, that is dumbbell. So these are asbestos bodies. They're also called as ferruginous bodies. When we say ferruginous bodies, probably something should strike to your mind. Ferruginous, ferrous, that means iron. So on these asbestos fibers, hemosiderin gets deposited. That's why it is like this. Can you recall how we can demonstrate that this is iron or it contains iron or this is hemosiderin? Any stain? I'll give you a clue. Blue, Prussian blue or pearl stain. So iron will take a blue color. So this is people who work in asbestos industries, they inhale these fibers, very small particles. So they evoke an inflammatory response there, a granulomatous inflammatory response, because it is something which is not local to that particular structure. It is recognized as a foreign body. So foreign body granulomatous reaction, fibrosis, everything. So it leads to asbestosis, a pneumoconiosis. So people who have asbestosis are prone to develop a malignancy, that is mesothelioma. So where does mesothelioma occur? It can occur in the pleura or in the peritoneum, because pleura and peritoneum have the lining of mesothelial cells, and these people with asbestosis can develop bronchogenic carcinoma as well. Okay, association of a pneumoconiosis and malignancy you have seen here. Please don't forget asbestosis, mesothelioma. Then probably few words about this. How do we detect the carcinogenicity of a chemical? How do we say that this chemical is carcinogenic? Means there are experimental models. I just want to stress on few points here because sometimes this could be a 3-marker for you. So what is done is the chemical compound under test or to be tested is administered to experimental animals. How is it administered? It all depends on what chemical we are testing. Is it a chemical that is known to produce carcinoma of the skin? Then it has to be painted. Okay. Is it a chemical that produces malignancy in vivo? Then either inhalation or ingestion or by parenteral route we have to administer this chemical. Okay. So then different doses and varying periods and all these we need to know how long it takes and all that. Then the animal is sacrificed and studied. Of course, it has got a lot of drawbacks. We can't say all positive or negative tests can be applied to humans. We can't say if it occurs in experimental animals, it occurs in the humans also. And this is actually mutagenicity testing of a chemical. The name of the test is Ames test. AME' test. What is a mutagen? It's a substance that can permanently alter the genetic composition of the cell. So what does this test do? Ames test evaluates, just know this, it evaluates the ability of a chemical to induce mutation. It evaluates the ability of a chemical to induce mutation in the mutant strain of Salmonella typhimurium. So that cannot synthesize histidine. See, for the test, there is a microorganism that is used, bacteria, Salmonella typhimurium. The speciality here is it's a mutant strain. That means it cannot synthesize histidine. Okay, just look at this test. The strain, I told you, mutant strain of Salmonella typhimurium is here. Then there's a conical flask where the suspended chemical carcinogen is there, and to this liver homogenate is added. Okay, so that will supply the enzymes which are required to convert procarcinogen to ultimate carcinogen. We are testing it. So liver homogenate. So liver homogenate and the chemical which is to be tested plus Salmonella typhimurium mixture is there now. So this is taken and added to a culture medium. This culture medium doesn't have histidine, so bacteria cannot grow there. So if this is a carcinogen, then this mutant strain will produce histidine and there are bacterial colonies that develop because the chemical under test is mutagenic. So it will induce mutation in the mutant strain of Salmonella typhimurium in the form of functional histidine gene. Okay. So this is reflected in the form of bacterial colonies. Thank you. So that's about part 2 of chemical carcinogens. I've just simplified this topic for you. Please read, as I told you, recollect, review, read. Okay. So take care. See you in probably in the next class. Bye.